Um, Hi everyone. I'm so excited to be here. My name is Soha Leem, and I am a rising sophomore currently working in Dr. Plekia's lab, along with the help of Dr. Baccio and graduate student Julia. Today, I will present to you my progress so far on trying to transform, express, and purify the FA2 receptor ligand binding domain with the alanine 158 methionine mutation. So quick introduction, in particular cancers such as breast and pancreatic, it was found that the FA2 tyrosine kinase receptors are overexpressed. This means that there must be some relation between the overexpression of the FA2 tyrosine kinase receptors and the metastasis of breast and pancreatic cancer cells. And fortunately, recently developed targeting agents such as 135H12 can actually be used to bind to the FA2 receptor and cause degradation, which allows for the reduction of FA2-driven cell migration. And pictured here is a tyrosine kinase receptor functioning normally. This type of receptor is activated through dimerization in the presence of ligands, which are the red dots pictured near the blue receptors. And when the ligand successfully binds, this results in the transduction of the signal and the corresponding cellular response. Which in the case of my experiment and the FA2 receptors is the suppression of cancer metastasis. So going off of this background information and very general introduction, an objective was formed for my project to perform a detailed analysis of the binding mechanisms between the ligand and the mutant FA2 receptor that carries the isotope, which will help illuminate a visual analysis on how to create binding agents that have a strong affinity towards the receptor. And more simply, if a detailed analysis of the binding mechanisms between FA2 and its ligand is conducted, this opens up the potential for investigating whether certain agents, like mentioned before, 135H12, could be verified or disproved. And to accomplish this objective, first transformation was performed. But just to give an idea of the transformation protocol and its relative significance, I have created a flowchart. And like a board game, the protocol starts on green and ends on red. So first, the competent cells were thawed in ice. It was gently mixed with plasmid, then cooled down with ice. Then it was placed on ice in heat shock so that the plasmid could be taken up by the cell and returned back to ice. Then the SOC medium, or the nourishment for the cells, was added to the tube, and the tube was shaken in an incubator so that the cells could be nourished and grow. The bacteria was placed in different concentrations onto lauryl broth agar plates containing ampicillin, and finally they were incubated at their optimal temperature overnight so that they can grow. And after successfully transforming the competent cells with the plasmid, the cells were expressed to start producing our desired mutant receptor. To express the bacteria, the condition that works for the wild type FA2 was used. So the glycerol stalk was scratched and used to inoculate a flask of LB media that contains ampicillin. Then it was incubated overnight at the optimal temperature. This pre-culture was then used to inoculate 1 liter of LB media. It was shaken until the optical density was 0.6 and then induced with IPTG. And before centrifuging, bugbuster solution was used to break down the cell membrane and allow the protein of interest to be released into the supernatant. And in doing so, we can ensure with gel electrophoresis that our protein was present and that the protein had been successfully extracted into the supernatant. The cells were then collected and centrifuged, and after the pellet was resuspended with lysis buffer and frozen at -80 degrees. And once it was successfully verified that our mutant receptor protein was successfully expressed, we moved on to the purification of the protein. And to purify, protease inhibitor and DTT, a reducing agent, were added to the thawed cells resulting from the expression protocol. And the resulting mixture was sonicated in on and off cycles to weaken the cell membrane and allow the desired protein to be released into the supernatant. Then the mixture was centrifuged, and after, to further purify the protein, the supernatant was filtered. And finally, IMAC chromatography and buffer exchange were performed to further purify the mutant protein. Now to the physical results. So first we tried the normal condition for the expression used for the wild type FA2, which was using LB broth and origami competent cells. But for this gel specifically, the bacteria was left at 20 degrees Celsius overnight after being induced with 0.5 millimolarity of IPTG, which is a chemical reagent that removes a repressor from the lac operon to induce protein gene expression. And as can be seen in the gel here, circled in red is our protein. And the fact that it's present in the pellet and not the supernatant means something in the expression didn't go so well and led us to believe perhaps maybe the bug buster solution didn't work, or that the protein was not soluble. This is the FPLC for the wild type FA2 protein. And to give the briefest idea of how FPLC works, the line steeps downward when the substance is making its way down into the tubes. So because the wild type protein was for the most part present in our mixture, there is a prominent second peak visible, which represents our protein. But keep in mind, this is just a test run, and what we're really trying to successfully express is the mutant version of this protein. And when we tried to purify the mutant protein with FPLC, we find out that it was not present in the supernatant, the absence of which I circled in red and put question marks. And because before doing FPLC, the membrane was broken down with sonication, it could be concluded that it wasn't the bug buster solution that led to faulty protein expression, because the sonication did lead to the same result of the protein not entering the supernatant. So what it looks like now is our protein is not present because it is not soluble. So now that the normal conditions for the wild type FA2 didn't work for the mutant, we tried to optimize the conditions. So overall, we optimized the conditions by changing the concentration of IPTG, changing the temperature, changing the time in which bacteria was expressed, and then testing them with the gel. And in this specific condition, the bacteria was left at 37 degrees Celsius for 4 hours after being induced with varying amounts of IPTG. But as circled in red, the protein was only present in the pellet and not the supernatant. In this specific condition, the bacteria was left at 16 degrees Celsius overnight, again induced with the same varying amounts of IPTG, but again, the protein was only present in the pellet. In this condition, we used Rosetta Gammy competent cells instead of origami, but again, the protein was only present in the pellet. In this condition, the same results occurred, unfortunately, but so it has been hypothesized due to all of these gel electrophoresis results that the reason why our protein is not entering the supernatant is from its insolubility due to its folding structure. So although my results may seem like a huge, massive failure, I learned it really isn't. Something I learned from all the RISE workshops is that it is results like these that act as sort of the foundation for a successful result. And to my surprise, I learned from a conversation with my graduate mentor that it could take 10 years and usually oftentimes even more to get even one working drug that alleviates the disease. So overall, we learned all the various conditions in which our mutant protein receptor cannot be successfully extracted, which obviously brings us a lot closer to understanding both the folding structure of the protein as well as the binding mechanisms between the receptor and its ligand. And as I continue to work on this experiment and optimize even more conditions, perhaps we could discover the potential for using the proposed agent as a possible therapeutic in combination with standard of care, where it can help suppress a major influence of tumor metastasis. So I'd like to give a very special thanks to my primary investigator, Dr. Plekia, for his mentorship and encouragement. Dr. Baccio for guiding me every step of the lab procedures, and Julia for inserting her much needed advice while also mentoring me through the process as well. I'd also like to thank CNAS alumni and RISE donors for their generous funding and providing direction on how to successfully pursue research, as well as giving amazing presentations, allowing me to proceed forth in my academic journey with a lot more confidence. Um, so that marks the end of my presentation. Thank you very much for listening. Now I'd be happy to answer any questions. Oh, and here are my references as well.